Hi, everybody. Welcome to JAX Tech Talk with the Technical Information Scientists at the Jackson Laboratory. My name is Dolores, and my role as a Technical Information Scientist is to serve the research community with education and practical solutions. JAX Tech Talk is a weekly 15-minute podcast-style webinar where we answer frequently asked questions. Today I have Dr. Lizzie Axton with me to talk about the diversity Outbred pilot grants funded by JAX and why looking at genetic diversity is an important aspect for translatable research.
Yeah, thank you for having me, Dolores, and thank you everyone who's here live. My name is Lizzie, and I am a technical information scientist alumni. Dolores made me wear this shirt today. I'm just going to put that out there. I'm currently the Opportunity Development Manager, specifically for Model Generation Services. So at this point, genetic diversity is a little bit of my extracurricular activity, but I've spent a few years working with JAX researchers on this topic, developing educational resources and helping researchers branch into this new field. So I'm happy to be here.
Great, great. For anybody watching live, please use the comment section of the video to tell us where you're viewing from or any related questions you may have, and we'll try to answer those today. So most people doing medical research know that not all treatments are efficient for all patients. The traditional practice of one-size-fits-all approach to medicine is based on broad population averages and often misses the mark because each person's genetic makeup is different from each other. So often in very important ways that affect human health.
Exactly right. Most treatments will work for some patients and not others. And on average, what we see is that a given prescription drug may only work in half of the patients that actually take it. And that's really only the successful drugs overall. There's a long list of drugs that were tested, in various different animal models, whereas when they get to the clinical trials, they either don't pass due to lack of efficacy or unforeseen, issues with toxicity. So it's a huge challenge, and we really need a way to improve the translatability of our animal research to make sure that it works in the clinic.
That's right. That's right. There have been a lot of advances recently promoting personalized medicine, also known as precision medicine, especially in the field of oncology. For example, tailoring healthcare to each person's tumor's genetic makeup. So, Lizzie, how are researchers in the drug development field incorporating genetic diversity as they screen and test candidate leads?
Yeah, so one reason why sometimes research done in animal models is not translatable is that oftentimes we test interventions in a single inbred strain. And that is essentially one observation, and it's similar to testing a therapeutic in a single human patient. Whereas our human population is incredibly diverse. We have people of different genetics and sexes and ages, and these people are all exposed to different environments. So these differences can determine an individual's predisposition to different diseases and their response to treatment. So instead of tightly controlling all of the experimental variables in our animal studies, I really think that we need to embrace the complexity that we see in human life and find a way to include that complexity in our animal research. And one way we can start to address this is by including genetic diversity, so that way we can at least see how different genetics impact these experimental outcomes. One example that is relevant to all of us right now, Nadia Rosenthal from the Jackson Laboratory published an example of the impact of genetics on individuals' response to COVID. And I think that is a really striking example of how genetic background impacts research outcomes. Dolores, do you maybe want to walk us through that research example?
Absolutely, absolutely. Before I do that, I just a shout out from a few of our viewers. We have Xing Tong from Davis. We have Dr. Olabord from Edinburgh. We have Janine from California, Angela from Maine. So welcome everybody. It's great to see you here. So going back to Nadia's research, as you all know by now, one of the hallmarks of COVID-19 is the variability in the disease progression and the response to infections. While SARS-CoV-2 causes severe fatal disease in some patients, most people are infected and remain asymptomatic. So other than certain comorbidities like diabetes, for example, our own genetic variability may underlie these differences in response, right? So, but this is very difficult to validate in human populations. So to explore the role of genetics in COVID disease, Nadia Rosenthal, a researcher at JAX, and collaborators, they teamed up to generate a diverse panel of mice analyzing their infectivity. And how they did this, they bred a well-known mouse model for COVID research. We've talked about it in other tech talks. It's a mouse that expresses human ACE2 receptor, and they bred it to 8 well-characterized strains. And then the hybrid transgenic progeny was infected with SARS-CoV-2 that carried identical viral loads, so they were all treated the same, and they were monitored for 21 days. And what they've seen is that the results recapitulated what you see in human patients. Basically, different mouse strains displayed different disease severity. And let me share my screen for a moment to show you an example of what we're talking about here. So as you can see in the survival curves, you do see my arrow. So here we see on the y-axis survival of the mice, and these are the dates after inoculation. And you can see that, for example, the background, genetic background C57BL6 is very sensitive to the virus. So they all die, males and females, more or less after a week of infection. And on the other hand, you have some other genetic backgrounds like PWK shown here, where yes, the mice get a little bit of hit, but for the most part, they don't die, right? So they are resistant to the same infection as C57BL6. And another interesting finding from Nadia is that for certain genetic backgrounds like the inbred strain cast, we see that there's a sexual dimorphism, meaning that the males get a lot more hit by the viral infection compared to the females, and that's something that also recapitulates what we see in the human population. So, like we said before, this research was done in different inbred strains, so hybrids of the transgenic ACE2 mice bred with different inbred strains. So Lizzie, what other strains can be used to not only look at susceptibility to disease, but also to find out if a potential therapeutic is robust enough to work in different genetic backgrounds?
Yeah, really good question. So there are a lot of different ways to incorporate diversity in your study. Starting with, like Nadia did, of including multiple different inbred strains, all the way through working with diverse populations of mice. Now, I can't possibly cover all of them today, so I will say that we recently published a diversity user guide that actually walks through all the different options and how to include them in your research. So definitely check that out on our website if you want to know more about this topic. The one that I'd really like to highlight today is the JAX diversity outbred mice. Now, this is a really unique option because it started with 8 different inbred strains, actually the same 8 ones that Nadia used in her research, and basically bred them together in a very non-scientific terms, every which way, many generations, until you end up with a diverse population of mice that has millions of randomly segregating variants. Now, these diversity outbred mice are currently the most diverse population that exists and has an estimated 50 million segregating sequence variants. And what this means is that basically each mouse is unique. They have different coat colors and sizes and shapes and behaviors. They are susceptible to different diseases and infections, and responds to therapeutic interventions differently. So they provide a really unique opportunity to really ask a bunch of different types of research questions in the context of a diverse population, similar to humans.
Right, right, right. So can you expand a little bit more on how researchers can use these diversity outbred mice to um, look at translatability?
Yeah, absolutely. So, what we started talking about here today was just the translatability crisis of, animal research in general. So one way that you could use diversity outbred mice to assess translatability is what we call the diversity arm study. So in this case, let's say the example you have, a bunch of research that you've done in a single inbred, B6J, for example. What you could do with diversity outbred mice is do just a single, follow-up study with a small amount of mice to see if your results will be recapitulated when you add diversity. Yeah, so in the figure that Dolores is trying to move her mouse around for me, basically if you have, an established baseline, in your inbred, you might see that, all of the values go, let's just say, down upon intervention. If you were to do this in diversity mice, there might be a different baseline, there might be a different scale of effect, but, you would hope that it would be at least the same direction and hopefully the same magnitude. In contrast, if you do this in diversity mice and see that the values are all over the board, some are increasing, some are decreasing, some are the same, that would indicate that the likely explanation is that genetics are playing a pretty significant impact in your intervention. And you might need to investigate that further to figure out what those genetic variables are and how they might impact your results when you get into the clinic. So it's really just I'll say, a quick and dirty way to do a small study to see if your inbred findings will also be seen, in a diversity option.
Right. So in the top graph, we would conclude that this treatment would be more translatable to humans because in different genetic backgrounds, it still has a similar effect, whereas in this one, it may be translatable for some patients, but not for all, right?
Exactly that. And it does kind of go back to the precision medicine point too that you were making, Dolores, is that, maybe in that case, that drug will just be effective for some people. It's not to say the drug won't work, but maybe just in some groups of people, it would be effective.
Right, right. Yeah, and we got a question from a viewer. What if I use diversity outbred mice and my results are very variable, like in the graph below here? Is it a good idea to publish that, or is that rubbish and nobody's going to accept it?
I think what I like about this study design is that there is no way to fail. Whether your results are recapitulated, great. That gives you some additional information to use to support go into clinical trials and getting additional funding for that. If they are variable, that also opens up other research avenues. For example, you could then do a larger scale study with these diversity outbred mice and investigate the genetic basis of it, figure out what genetic variants are driving that difference. So it could actually also be a really valuable source of information. So I really think there's no way to go wrong with this. And whatever results you get, it's useful to publish, and also just even for, the broader scientific community to see, the use of these mice and how that impacts things. So.
Right,
Yeah.
right. So this brings me to Janine's question that I posted here for everybody to see. Basically, it's like, okay, you see that there's variability. How do you follow that? How do you follow up on that? And her question is specifically, has Dr. Nadia Rosenthal, has gone into what is driving these differences in response or not? I don't know if you have any inside information about what's her follow-up research on that or not.
Yeah, definitely. I know that is sort of the broader plan of our group is to do further analyses and figure out what precisely are the genetic variants, driving those differences. Honestly, the biggest way that you could do that is with something called QTL mapping. And that is actually what diversity outbred mice were originally developed for. I think the one thing I'll say is with the diversity arm approach that we just talked about, it's very small studies that wouldn't be sufficiently powered for a QTL mapping study. So you would have to do a follow-up study that's more larger scale to be able to accomplish that. So you need numbers for that, and you also need to make sure that you're working with the right mice.
Great. Thank you so much for that. So we also wanted to take the opportunity today to feature the release of a diversity outbred pilot grant program that is funded by the Jackson Laboratory. So Lizzie, can you walk us through the highlights of this grant? What type of research do they fund, and what are the deadlines?
Of course. Yeah, no, I'm really excited about this grant program. It's really the first time JAX has done something like this. And, everything that we've been talking about so far today, promoting translational research, incorporating genetic diversity, this is really designed to support that for the broader biomedical community. So with this grant, you can get basically up to $10,000 in basically direct cash for your research, and up to 50 free DO mice to support your studies. Because we are a nonprofit institution, this is only for researchers who are affiliated with academic, government, or nonprofit institutions in North America. It covers all therapeutic areas. We showed you the example of the study arm type of study. You can do that with really any therapeutic area that you're working with. And also, if that doesn't fit your goals, I'm really curious to see, what people come up with of how else they can work with DO mice in their research. So I would say it's pretty open bounds in terms of what you come up with and how to use DO mice. We are requiring that you share your results. Now, this doesn't necessarily need to be a formal publication. It can also be a poster or an abstract, really any way that you're getting your research outcomes out there into the community, because that really is essentially the whole goal of this. The application itself is relatively straightforward. We kept it pretty simple. You just need a project summary, a short research plan, and the usual suspects of budget and biosketch. And there is more information on the application process on the funding announcement, which I believe we sent out links to. Applications are now open as of last Friday. They are due December 3rd, and then you will be notified of the awards starting, I believe, January 14th is when those announcements will go out. So overall, like I said in the last slide, these are designed to be, not huge studies to do QTL mapping. It's really just to see what you can do with a small amount of mice to support research that you already have ongoing in your lab. And we hope that this is an avenue to sort of get people started with diversity, get people talking about it and starting to incorporate it into their labs and see what they can do.
And share the results, right?
Exactly, and share the results.
Right. Great. Well, that's all the time we have for this week. Lizzie, would you like to go over the links that we have posted for people in the description to find out more about these topics?
Yes. So in the description, we included a couple of different links for you for more information. One of them is the strain data sheet for the diversity outbred mice. I glossed over that very quickly, but that data sheet has a lot more information on those mice and how to work with them. We put in a link for the grant funding announcement. So that has all the information that you'll need to get started with that. And then finally, we also put in a link that is for our research facility and the Genetic Diversity Initiative. And that has a lot of helpful information on diversity mice and how to work with them. So I definitely recommend utilizing those resources in terms of planning your studies. And of course, always feel free to reach out to TIS, micetech@jax.org for questions about, working with diversity or mice in general.
Great. Thank you so much. Well, thank you everybody for joining us this week. Our next JAX Tech Talk is called Precannulated Mice and Other Surgical Services that we offer at the Jackson Lab. This will be live Tuesday, October 19th. And follow JAX Tech Talk on LinkedIn so you're notified anytime there's a new episode, or you can also subscribe to our playlist in YouTube. So this is Dolores saying stay healthy, stay safe, and stay excited about your research. Bye, everybody.
Thank you, everyone. Bye.
Thank you, Lizzie, so much for participating.